Everything most people know about a neurodivergent brain is framed as absence, the attention that doesn't hold, the social world that requires conscious construction step by step, the sensory environment that arrives at full force, unfiltered. Decades of clinical language produced a catalogue of what these brains cannot do. That catalogue is real. It is also incomplete. The same brain that finds an ordinary classroom physically overwhelming can detect the single mistuned string in an orchestra of 40 musicians before the conductor does. The same sensitivity is doing both. The same brain that finds the unwritten rules of a social gathering genuinely opaque can reconstruct every word of a conversation that happened 4 years ago in precise sequence. The same brain that cannot hold a grocery list in mind for 3 minutes can spend 6 hours mapping the internal logic of a system it finds interesting without being asked and without stopping. These are not consolation prizes for the deficits. They are the same architecture, seen from 2 different directions. The brain we are spending tonight inside is not a broken version of the standard model. It is a differently ordered one. The same developmental program that makes certain environments genuinely costly also produces a processing depth that the standard brain cannot reach by the same route. Vary the program, and you do not get a faulty brain. You get a different one. The word for this was coined not in a clinic but in a sociology thesis by an Australian researcher in the late 1990s. She noticed that the conditions being called disorders had something the clinical frameworks were not set up to see. They clustered in families and ran together in the same individuals. They came with genuine costs in certain environments and genuine advantages in others. They were not individual failures. They were variations in how a brain could be organized. The human brain contains around 86 billion neurons. Each one may connect to thousands of others. The total number of connections in a single brain exceeds the number of stars in the Milky Way, and every one of those connections was established according to a developmental program encoded in the genome. Vary the program, and you vary the brain, not randomly, in recognizable, heritable, consistent directions. Roughly 1 in 5 people has a brain that fits somewhere on the neurodivergent spectrum. If this were simply malfunction, something that reduced survival and left nothing in its place, it would not have persisted at these numbers across every human population ever studied. What makes a brain process the world at a different frequency from the one most social environments were built for? Why does the same underlying architecture that creates friction in certain contexts also create a depth that brains without it cannot easily reach? And why do these differences cluster together so that a brain wired differently in one way turns out to be wired differently in several? If you enjoy spending time inside questions like these, a like would be highly appreciated. And if something in what follows raises a thought or a question you want to put somewhere, the comments are exactly the right place for it. Now, let's begin. Neurodivergent was not a word coined in a laboratory. It did not come from a clinical trial or a diagnostic revision. It came from a sociology thesis, written in the late 1990s by an Australian researcher named Judy Singer, who was studying something specific: how society constructs the categories it uses to describe minds that do not fit the expected pattern. Singer noticed that the conditions her research was examining had something the standard medical model did not account for. Autism, ADHD, and dyslexia were not randomly distributed across the population. Neither was dyscalculia or any other condition gathered under the same clinical umbrella. They clustered, not just in individuals, but in family lines across generations, appearing together in the same children and the same parents and the same siblings in proportions that chance alone could not explain. They came with specific difficulties in specific environments and specific capabilities that neurotypical minds often could not replicate. And they had been present in every human population ever studied at roughly the same frequencies across time. Singer called this range of variation neurodiversity. It was a deliberate choice. Diversity, in biological terms, refers to variation within a species that is not simply malfunction. A species with low genetic diversity is a fragile species. Variation is the raw material of adaptability. Singer was proposing that what the clinical frameworks had been treating as a catalogue of disorders was better understood as a range of brain configurations, each with particular characteristics, each representing a different set of trade-offs between processing strengths and processing costs. The word did something that clinical language had not managed. It separated the idea of difference from the idea of deficit. You can be different in how your brain processes the world without that difference being reducible to a list of things you cannot do. The deficit model, which had dominated clinical practice for most of the 20th century, defined these conditions entirely through what was absent or impaired relative to a neurotypical standard. Neurodiversity proposed that the standard itself was a statistical construct, not a biological ideal. To be clear about what this is and what it is not, framing neurodivergence as variation is not a claim that these conditions cause no difficulty. They often cause significant difficulty, and flattening that is its own kind of error. A person who cannot process a standard school environment is genuinely impaired within it. One who cannot hold a job structured around neurotypical expectations, or who cannot manage sensory overload in a public space, faces real barriers. The variation is real, and the mismatch between the variation and the environment is also real. Both are true simultaneously. What the neurodiversity framing offers is a different question. Not, what is wrong with this brain? But, what is this brain actually doing, and why does it do it this particular way? The neuroscience that has accumulated in response to that second question is what makes the story worth spending time with. The conditions grouped under the neurodivergent umbrella are clinically distinct. They are diagnosed by different criteria and described as involving different mechanisms. But at the level of the brain itself, at the level of how these brains develop and how they are wired, they are far less distinct than the categories suggest. The clinical lines are drawn around behavior. The brain does not honor those lines. And here is where the clinical account and the neuroscience begin to diverge in a way that matters. When a clinician diagnoses autism, they are identifying a cluster of behavioral traits. The traits include differences in social communication, sensory processing, and the intensity of interests. When a neurologist scans an autistic brain, they do not find a broken region or a missing structure. They find a different pattern of connectivity. They find a brain that has not failed to develop the standard architecture. It has developed a different one. The history of how these conditions came to be called disorders is worth a moment. For most of the 20th century, autism was understood through a lens of absence. The early clinical descriptions, written in the 1940s by the psychiatrist Leo Kanner, framed autism as a failure of social attachment. Children who did not make eye contact, who did not respond to their name, who seemed unreachable. The framework was designed around what these children could not do. That framework remained dominant for decades. It was not a stupid framework. It captured something real about the difficulties involved. But it was designed to describe the problem, not to explain it. And because it only ever looked for what was missing, it was slow to notice what was present. Neurodiversity did not replace the deficit framework with something softer or less accurate. It replaced it with something more precise. Not what is absent, but what is actually here and how does it work? The neuroscience that followed from that second question is a different kind of science. It is science that finds things. Understanding what that different architecture actually looks like and what it produces when it runs is the thread that pulls through everything that follows. When brain scanning technology became available in the 1980s, researchers turned it toward autism, expecting to find something they could point to: a lesion, a missing region, tissue that had failed to develop in the usual way. The search made sense given the model. If autism produced specific impairments, those impairments should be traceable to specific anatomical absences. The search found nothing consistently there to point to. Autistic brains had a cortex and a hippocampus and a cerebellum. The regions were roughly where they were supposed to be. The tissue looked like tissue. Early studies came back with findings that contradicted each other because there was no single absence that appeared across enough people to constitute a signature. The search for the broken part kept turning up a brain that looked structurally like a brain. This was genuinely puzzling. The behavioral differences were real and consistent enough to be diagnosed reliably across clinics and cultures and generations. But the anatomy kept refusing to supply a clear explanation. The resolution came from looking not at what the brain was made of, but at how it was talking to itself. A cognitive neuroscientist named Marcel Just, working at Carnegie Mellon University through the late 1990s and into the 2000s, began mapping which brain regions activate together during specific tasks in autistic compared to non-autistic people. He was not looking at individual regions in isolation. He was looking at the functional relationships between regions, the degree to which one region's activity predicted another's at the same moment, and the network-level organization of how a brain coordinated itself across a task. The findings were consistent across task after task. When autistic participants performed verbal tasks or social reasoning tasks, their brain regions showed a different pattern of coordination. Within any single processing area, the local connections were often enhanced. Nearby regions spoke to each other richly and reliably, but the long-range connections between distant brain regions that normally coordinate complex integrated behavior were quieter, less synchronized, less active across the same tasks. Just and his colleagues published these findings and described the pattern as underconnectivity. That word has caused some confusion. The autistic brain is not underconnected in the sense of being sparse or impoverished overall. Within its local territory, it may be more richly connected than average. The underconnectivity is specifically in the long-range links, the ones that normally integrate information across different processing systems simultaneously. The brain was not missing anything. It was wired with a different balance between local richness and long-range reach. This distinction matters because local richness and long-range reach produce different things when they run. A brain that processes information with unusual density within any given domain will do certain things exceptionally well. A brain optimized for rapid integration across many domains simultaneously will do other things exceptionally well. When the balance shifts toward local richness, you get a brain that processes detail with unusual depth and consistency. You also get a brain that finds it harder to coordinate multiple simultaneous information streams, which is precisely what complex social interaction demands. A conversation requires simultaneously tracking what is being said and what was said before it. It requires reading the other person's face, judging the right emotional register, and forming your own response. All of this arrives in parallel at the same time as a single event. The more you need to integrate across those streams at once, the more the reduced long-range connectivity costs you. This is why the autistic experience of social interaction is not simply less enjoyable than the neurotypical one, it is structurally more difficult. Not because social understanding is absent, but because the architecture that would integrate all the arriving signals simultaneously is calibrated differently. The significance of Just's findings extended beyond the imaging lab. Connectivity research changed what questions researchers asked. If the brain was not missing pieces but had a different wiring pattern, then the relevant questions shifted. Not, where is the damage, but how does this different wiring produce the behaviors that clinicians were observing? How does reduced long-range connectivity translate into the specific social, sensory, and cognitive profile that autistic people report experiencing? Those questions opened up entirely new lines of investigation. Researchers began studying functional networks rather than individual regions. They began comparing how whole brain coordination changed across different tasks, rather than looking for single broken components. And the more they studied networks rather than regions, the more consistent the pattern became. It also raised a deeper question that researchers are still working through. If the wiring is different, but the pieces are all there, where did the different wiring come from? Something during development set the local connectivity high and the long-range connectivity relatively low. What was that something, and when did it happen? The question is what that excess of local wiring actually produces in the brain that has it. What does a brain with dense local connections and reduced long-range ones actually do differently? Think of a country's telephone network built with extraordinarily dense local lines. Every street in every neighborhood connects richly to every other street in that same neighborhood. Inside any single city, conversations flow with speed and detail, but there are fewer trunk lines running between cities. Information traveling across the country must take longer routes, pass through more exchanges, and loses some of its richness along the way. That is a rough approximation of the connectivity pattern researchers keep finding in autistic brains. Within any one processing area, the local connections tend to be denser and more elaborate than average. More cells communicating with nearby cells, more intricate local organization within any given region. But the connections that would typically link distant regions together, the long cables that allow one part of the brain to instantly share information with another part far away, are less active and less synchronized. This pattern has a name now: enhanced local connectivity with reduced long-range connectivity. It was identified across multiple imaging techniques and dozens of studies, and it shows up with enough consistency to be treated as one of the most reliable structural signatures of autism yet found. What does enhanced local connectivity actually produce? Consider vision. When the brain processes visual information, it first handles fine detail at the local level. Edges, textures, and color contrasts within a small region of the scene are resolved before anything else. Then long-range connections carry that local information upward and outward, integrating it with information from other regions until the brain constructs a coherent interpretation of the whole. More local connectivity means the fine detail stage is handled with unusual richness. But if the integration stage is relatively quieter, the brain arrives at the whole by a different route. It may notice details that a less locally connected brain would smooth away. It may also take longer to arrive at the gestalt, the overall interpretation, because that requires more long-range coordination to complete. This is not a deficit, it is a different path through the same problem. The psychologist Uta Frith, who spent decades studying autism at University College London, developed a framework she called weak central coherence to describe something she kept observing in autistic people. the tendency to process parts before wholes, to notice the individual triangles in a pattern before the shape they make together, to hear individual notes before the melody. She initially framed it as a cognitive weakness, a failure to integrate. Over time, the framing shifted. The central coherence that was weak was not intelligence or attention. It was the automatic pull toward the gestalt that most brains apply without effort or choice. Autistic brains were not failing to integrate. They were doing something different with the local information before integration happened, and that something different turned out to be a genuine advantage in any task where the local information was what mattered, finding an embedded figure hidden in a complex design, detecting a pattern break in a long sequence, noticing the single anomalous note in a melody played by an orchestra. On tasks like these, autistic participants not only performed as well as non-autistic ones, they performed faster and more accurately. The same connectivity that made the gestalt harder to reach made the detail easier to see. Beyond vision, the pattern shows up in auditory processing too. Autistic people often show unusual facility with pitch. They detect small differences in frequency that fall below the threshold of reliable detection for neurotypical ears. They are more likely to have absolute pitch, the ability to identify a musical note without a reference, which appears in the general population in roughly 1 in 10,000 people, and in autistic populations at rates many times higher. This is not because autistic people learn music differently. It is because the auditory cortex is processing fine spectral detail at a resolution that the standard calibration does not reach. The same pattern appears in language processing. Autistic people often show unusual sensitivity to the phonetic details of speech, individual sounds, and their acoustic properties, while finding it harder to automatically extract the social and pragmatic meaning that neurotypical brains derive from tone and rhythm and context. The local level is processed richly. The integration across levels, from phoneme to word to social meaning, is done differently. Uta Frith called this detail-focused processing, and she spent years arguing that it was not a failure of global processing, but a genuine processing style with its own coherent logic. The autistic person who notices every leaf on a tree before arriving at the tree is not doing less than the person who sees the tree first. They are doing something different, at a different resolution, across a different time scale. What is harder to explain from the connectivity account alone is why these processing differences produce such specific perceptual effects. The answer requires understanding something more fundamental about what the brain is doing with incoming sensory information before any of this processing begins. To understand why the connectivity pattern produces what it produces, you need a model of what the brain is fundamentally doing at every moment. And the model that has become increasingly central to how neuroscientists understand the brain over the last 2 decades is this. The brain is not a passive receiver of the world. It is a prediction engine. Before any sensory information arrives, your brain has already formed an expectation of what it will receive. The signals that arrive through your eyes, your ears, and your skin are noisy and incomplete. They are not high-resolution feeds. They are sketches. Your brain's job is not to record them faithfully. Its job is to combine what arrived with what was already expected and produce the best available model of what is actually out there. This process runs constantly below conscious awareness. Your brain generates a prediction, the world arrives. The gap between the two, the difference between what was expected and what actually came in, is called the prediction error. That prediction error is the only signal that gets passed upward for attention and learning. If the world arrives exactly as expected, there is nothing to update. If it deviates, the deviation is what the brain pays attention to and learns from. Think of a camera with a full automatic mode alongside a camera on manual settings. The automatic camera has learned what most scenes look like. Faces should be sharp. Backlighting should be compensated for. Skies should be correctly exposed. It applies those stored models to the incoming image and produces a handled, adjusted result. The manual camera captures what is actually there without correction. Both produce accurate images, but they produce them differently, and they fail on different kinds of scenes. A brain with strong top-down predictions works like the automatic camera. It smooths incoming sensory signals using everything it already knows about how the world usually is. Small deviations from expectation get absorbed into the prior model rather than reaching full awareness. The world feels familiar and manageable because the brain is constantly interpreting it through a filter of what it already knows. Now consider what happens when the balance shifts, when the brain weights incoming sensory signal more heavily than prior expectation, when the prediction does less of the work and the raw data does more. Carl Friston at University College London has developed frameworks for predictive processing that have shaped how the field thinks about brain function. Working alongside researchers including Pawan Sinha at MIT, these frameworks suggest the autistic brain may operate closer to the manual camera end of this spectrum, not because the predictions are absent, but because the weight assigned to them is different. Incoming sensory signals are trusted more, prior expectations are held more loosely, the world is met more directly. This has measurable consequences. A brain that trusts its raw sensory data more than average will notice more. It will be less likely to smooth away a deviation from expectation that the prior model would otherwise absorb. It will detect subtlety that a more prediction-heavy brain would miss, and it will find the continuous arrival of unsmooth sensory data more demanding to process, because every signal that would have been dampened by a prior expectation now needs to be handled at full amplitude. There is one further consequence of weighting raw sensory data more heavily than predictions, one that has received considerable attention in autism research. The behavior often described as repetitive or stereotyped. From outside, repetitive behaviors look like habits taken to an unusual extreme. Rocking and hand movements, repeated phrases and elaborate daily routines, deep focus on specific topics. They have been listed in diagnostic criteria as symptoms, treated as things to reduce, managed as obstacles to social integration. The predictive processing framework suggests something different about where they come from. In a brain that does not smooth incoming sensory variation with top-down predictions, the world is noisier, more variable, harder to anticipate moment to moment. Repetitive behaviors introduce predictability. They are self-generated sensory inputs whose pattern is already known, so the prediction error is zero. The brain gets a rest from the continuous effort of processing the unexpected. The behavior is not maladaptive, it is regulatory. It is the brain creating the predictability the environment is not providing. This reframes what an intervention that eliminates repetitive behavior is actually doing. It removes a regulation strategy from a brain that needs one without necessarily providing a replacement. The behavior looked like the problem. The unpredictability was the problem. This is not a flaw in the prediction system. It is a different parameter setting. And when you change the parameter, the sensory world changes with it. Think of a smoke detector set to a higher sensitivity than the standard. The one that triggers every time toast is made is not a broken detector. It is set to detect at a lower threshold than usual, calibrated for environments where the cost of missing smoke is higher than the cost of occasional false positives. Change the environment, and that calibration becomes a liability. Keep the environment right, and it is an advantage. The autistic sensory system is calibrated differently, not in one modality but across many, and not in the same direction or to the same degree in every person. But the pattern the research points toward is one of higher baseline sensitivity, a lower threshold before signals reach conscious awareness, less top-down dampening of what arrives. For many autistic people, this means that the world arrives at a different volume. A fluorescent light that a neurotypical person might not consciously notice is generating a real signal, audible as a faint hum at a specific pitch, visible as a flicker at a particular frequency. The seam of a sock against the skin is not a background sensation that fades into irrelevance. It is a continuing signal that the brain has not been able to classify as unimportant and set aside. The smell of a canteen or the temperature of a room that is 2 degrees off comfortable, these arrive fully without being filtered away. The sound of someone chewing across a quiet space registers as a signal the brain cannot easily deprioritize. This is not imagined or exaggerated. The sensitivity is real, and it has a neurological substrate. Studies measuring how quickly sensory signals travel through the nervous system in autistic individuals show processing speeds that differ from neurotypical populations in specific ways. Electrophysiological work measuring the brain's electrical response to sounds and images shows that autistic brains generate early sensory responses, the first electrical spike that marks the arrival of a signal, that are larger and more consistent than those seen in non-autistic brains. The signal is arriving with more force. The brain is registering it more strongly. The consequences compound in environments that were not designed with this calibration in mind. A standard school classroom combines bright overhead lighting with the ambient noise of 30 people breathing and shuffling. It adds the texture of chairs and uniforms, and the competing sensory streams of multiple subjects across a school day. For a brain receiving all of this at full amplitude, without the same degree of top-down dampening that reduces the cost for neurotypical students, the cognitive load is not equivalent. The learning task is running alongside a constant effort to manage a sensory environment that was built for a different calibration. The effort is real, and it has a metabolic cost. This is not a metaphor. The brain consumes energy. Sustained sensory management draws on the same resources as thinking and regulating and attending. A brain that is working harder on sensory management has fewer resources available for the tasks the environment is simultaneously asking it to perform. One further aspect of the sensory account is worth spending time on, because it is the one most often overlooked: interoception. The brain does not only process external sensory information, it also processes internal signals. The state of the body itself arrives at the brain as a continuous sensory stream, just like vision and hearing. Hunger and pain are signals the brain receives and must interpret. So are temperature and fatigue. That internal channel is subject to the same calibration differences. Many autistic people find it genuinely difficult to read their own body states. Hunger does not arrive as a clear signal that something needs addressing. It arrives late or masked behind other sensations. Pain follows a similar pattern, sometimes under-registered in ways that allow injury to go unnoticed, sometimes over-registered in ways that make minor discomfort genuinely disabling. Fatigue accumulates without clear internal signaling that rest is needed, until the deficit is large enough to produce a crisis. This matters because many of the behaviors that look like poor self-management in neurodivergent people are actually the consequence of imprecise access to internal state information. The person who does not eat until they collapse was not ignoring hunger. The hunger was not arriving clearly. The person who pushes through exhaustion to the point of breakdown was not being reckless. The fatigue was arriving without its usual urgency. The external world and the internal world both run through the same sensory calibration system. When the calibration is different, both are affected, and the sensory difference is only one part of the account. There is another difference, perhaps even more pervasive, in how the world of other people arrives. The most persistent claim made about autistic people, the one that has shaped clinical diagnosis and public perception for decades, is that they lack empathy, that they move through social situations without registering the emotional states of others, that their social difficulties are a consequence of emotional blindness. The neuroscience does not support this. In 1985, a researcher at University College London named Uta Frith developed what became known as the false belief test, working with colleagues Simon Baron-Cohen and Alan Leslie. The test was elegant. A child watches a character place an object in a box and then leave the room. While the character is away, a second character moves the object to a different location. The first character returns. Where will the first character look for the object? Most children above the age of 4 answer correctly, in the box, because that is where the first character believes the object to be, even though it is no longer there. They understand that another person can hold a belief that differs from reality. They have what psychologists call theory of mind, the ability to model what is happening inside someone else's head. Autistic children, Frith and her colleagues found, tended to answer differently. They pointed to where the object actually was, not to where the first character believed it to be. They were not lying or guessing. They simply seemed not to model the first character's belief as a separate internal state, distinct from the facts. This was taken as evidence that autism involved an impaired theory of mind, a difficulty in building mental models of other people's inner lives. For years, this framing shaped the field. Autistic social difficulty was understood as arising from an inability to empathize, a blind spot for what others were thinking and feeling. Then a psychologist named Damian Milton proposed something different. In 2012, he published a paper arguing that the test itself was asymmetric. The false belief test measured how well autistic people could model non-autistic minds. It did not measure how well non-autistic people could model autistic minds. One side of the mutual understanding problem had been tested, the other had not. When researchers eventually ran the experiment in both directions, something clarifying happened. Non-autistic people, asked to predict the behavior of autistic people, struggled as much as autistic people struggled to predict the behavior of non-autistic ones. The mutual understanding broke down in both directions. The failure was not a deficit in autistic brains. It was a mismatch between two different social operating systems. Milton called this the double empathy problem. The breakdown in social connection was not one-sided. It arose at the interface between two differently calibrated minds, neither of which found the other fully readable. There is further evidence. When researchers brought groups of autistic people together, without any non-autistic people present, something changed. The social coordination that was absent in mixed group interactions appeared. The warmth and mutual understanding were present when the cognitive style was shared. The easy exchange of shared frames of reference appeared without effort. Autistic people, talking with other autistic people, showed no particular deficit in social connection at all. The empathy was not missing. The decoding was difficult because the code was different. The double empathy problem also explained something that had puzzled researchers for decades: why autistic people often communicate better with each other than with non-autistic people. If autism was simply an impairment in social cognition, autistic people interacting with each other should show the same or greater difficulty than autistic people interacting with non-autistic people. The deficits would compound rather than cancel. The opposite is what actually happens. Autistic people interacting with other autistic people show stronger rapport and more successful information transfer than when either group interacts across the interface. The mutual understanding was there. It required a shared cognitive style to surface. This was demonstrated directly in a study by Catherine Crompton and colleagues published in 2020. The mismatch was not symmetric in either direction, but the autistic condition was markedly smoother than the mixed condition. This has a clear implication. The problem was never a one-sided impairment. It was an incompatibility between two different systems, each of which worked well on its own terms and produced friction when asked to decode the other. The clinical framing that focused entirely on the autistic side of that interface missed the other half of the equation entirely. Understanding this changes what social support for autistic people should look like. Supporting someone to navigate a world built for a different cognitive style is different from fixing a broken social brain. One of those approaches starts from deficit, the other starts from mismatch. The neuroscience now clearly favors the second. Many neurodivergent people develop a skill they are rarely taught and often cannot name. They learn, early and usually without being told how, to produce a version of themselves that fits more smoothly into the environments around them, to make the eye contact that feels unnatural, to produce the expected facial responses at the expected moments, to suppress the physical behaviors that bring unwanted attention, to filter the incoming sensory environment actively rather than letting it arrive at full force. This practice is called masking, and it has a cost that most accounts of neurodivergence do not include. Think of someone conducting all of their professional conversations in a language that is not their own. They may be entirely fluent. They may be indistinguishable from a native speaker to most people they meet. The content of every conversation is handled correctly. The tone is calibrated. The idioms are right. But running alongside each exchange is a second process, matching incoming phrases to meaning, selecting responses and double-checking register. Everything that a native speaker does automatically must be done consciously. By the end of a day of conversations in that second language, the fatigue is not social. It is the fatigue of sustained translation running in a system that was designed for something else. Masking works the same way. The social interaction succeeds, but behind the performance, a continuous translation is running between the natural behavior of this brain and the behavior the environment expects to receive. The resources that translation uses are the same resources available for everything else. Thinking, learning, and attending all draw from the same pool. Brain imaging studies have begun to quantify this. Autistic people who are actively suppressing their natural behavior during social interaction show different neural activation patterns from those who are not masking. The suppression is metabolically visible. The prefrontal regions involved in monitoring and controlling behavior are more active. The cost of the performance is real, not metaphorical, and it accumulates across a social day in the same way that physical exertion accumulates across a physical one. The consequences are predictable. A neurodivergent person who spends most of their waking hours masking across every social context that rewards neurotypical presentation is running a sustained deficit between energy expenditure and recovery. The depletion eventually reaches a threshold. What follows is sometimes called autistic burnout, and it is clinically distinct from ordinary fatigue, or even from the burnout that comes from overwork. Autistic burnout involves a collapse in the capacity to function that can persist for weeks or months. It is not tiredness. It is a withdrawal of the systems that support basic functioning. Language becomes harder. Sensory sensitivity increases. Executive function drops sharply and the capacity to mask disappears entirely. The brain is not failing. It is refusing to continue supporting a performance it no longer has the resources to sustain. There is a demographic pattern in this that took research a long time to explain. For decades, autism was diagnosed at much higher rates in boys than in girls. The assumed reason was that autism was genuinely more common in males. The actual reason, which the data eventually made clear, was that masking in girls and women tends to be more effective. Girls are more likely to have studied the social environment carefully and calibrated their performance to it. The suppression is more complete. The presentation under observation looks less distinctive, so the diagnosis does not come. It comes later, sometimes decades later. And for many women who receive a diagnosis in their 30s or 40s, what follows the relief of having an explanation is something else. A retrospective account of a lifetime spent working harder than the people around them to do things those people found natural, while assuming the difficulty was a personal inadequacy. The masking was not recognized as masking, it was experienced as effort, and the explanation for the effort was never the right one. The late diagnosis does not simply provide a label, it reframes a life, and for many people, that reframe is the thing that finally allows the masking to be reduced. Not because the external environment has changed, but because the person now knows what they are doing when they mask, which is the first step toward doing it less. What makes masking especially costly is that it is often invisible even to the person doing it. It develops so early and becomes so habitual that it no longer registers as effort. The performance becomes the identity, and the exhaustion it generates gets attributed to anything except its actual cause. The reason the same pattern appears across so many different conditions is that those conditions share more underneath their clinical labels than any individual diagnosis implies. The conditions grouped under the neurodivergent umbrella are diagnosed separately. They appear in different diagnostic manuals and are handled by different clinical specialists. From outside, they look distinct. Autism involves social and sensory differences. ADHD involves attention and impulse regulation. Dyslexia involves written language processing. The categories feel like separate problems with separate origins. Inside the genome, they are less distinct. Large-scale genetic studies conducted across hundreds of thousands of participants over the last decade have found something that the behavioral categories do not predict. The same gene variants appear repeatedly across multiple neurodivergent conditions. A person carrying a variant associated with autism has a meaningfully elevated probability of also meeting clinical criteria for ADHD. Someone with dyslexia shares more genetic landscape with someone with dyscalculia than either shares with the general non-dyslexic population. ADHD and autism share a significant proportion of their genetic risk factors with each other. The overlap runs deeper than the clinical categories suggest. This pattern has a name in genetics: pleiotropy, a single gene variant with effects on multiple traits simultaneously. The same gene doing more than one thing in more than one system. Think of a species of plant that carries a variation in the enzyme responsible for pigment production. That single enzymatic variation simultaneously affects the color of the plant's flowers, the shade of its bark, and the reflectance of its leaves. Each effect appears in a different structure. Each manifests differently. A botanist studying only flowers would call it a flower colour condition. A botanist studying only bark would describe a separate bark condition. Only when you look across many plants at once and notice that the flower variation and the bark variation always arrive together does the single origin become visible. Neurodivergent conditions work like this. The gene variants associated with them tend to be involved in how neurons develop, how synaptic connections form, and how the developmental balance between local and long-range brain connectivity gets calibrated. These are not condition-specific mechanisms. They are foundational processes that affect the whole developing brain, and their effects appear wherever those processes matter most. The genetic architecture is more complex still. No single variant produces neurodivergence. The architecture is polygenic, meaning that hundreds of genetic variants each contribute a small piece of the overall risk. No single one of them determines the outcome. The effect of each individual variant is too small to detect on its own. It takes large studies comparing the genomes of hundreds of thousands of people to detect the signal at all. The variants that matter are not rare mutations. They are common variants, present in the general population, whose cumulative effect in particular combinations shapes the developing brain in the characteristic directions that show up as neurodivergent traits. This polygenicity has a practical consequence for how we understand the conditions themselves. There is no single genetic test for autism or ADHD because there is no single gene. The risk is distributed. The same variants that appear at higher frequency in autistic populations also appear in their non-autistic family members, often at nearly the same frequency. What differs is not the presence or absence of the variants, but the dosage and combination. The traits that show up as diagnoses in some people appear in milder or subclinical form in many more. The genetic architecture produces a continuous distribution. The diagnostic categories are thresholds drawn across it. This also explains why the co-occurrence of neurodivergent conditions is the rule rather than the exception. Something between a third and a half of autistic people also meet criteria for ADHD. A significant proportion of people with ADHD have dyslexia or dyscalculia. These are not coincidences. They are predictable consequences of a shared genetic architecture whose effects do not stay within the lines the clinical categories have drawn. The clinical categories are useful. They help direct support toward the right kind of intervention, but they are maps of behavior, not maps of biology. The biology underneath them is more unified, more shared, and more continuous across the spectrum than any individual diagnostic label implies. And the genetic architecture that produces all of this does not assemble itself all at once. It unfolds during development, across months and years. And during that unfolding, the brain takes different shapes in different people along different timelines for reasons that are now beginning to be understood. No brain arrives complete. The brain you have now is not the brain you were born with, and not the brain you will have in 10 years. Development is not an event. It is a process that unfolds across years. The shape of the adult brain is determined by decisions the developing brain makes during those years, which connections to strengthen, which regions to expand, and at what pace to prune the rest. For most people, this developmental process follows a characteristic timeline. The brain prunes roughly half of all its synaptic connections between infancy and early adulthood. This sounds like loss, and in one sense it is. But the pruning is selective. The brain keeps the connections it has been using and removes the ones that have been firing less reliably. It is sculpting itself toward efficiency in the environments it has been experiencing. By the time development is largely complete, in the mid-20s, The adult brain reflects the accumulated effect of years of experience filtered through the genetic program it started with. In neurodivergent brains, this developmental process follows a different schedule and a different pattern. In autism, the first 2 years of life show accelerated cortical growth. The surface area of the cortex expands faster than typical, adding connections at a higher rate than the standard program would produce. Then, in early childhood, the pruning that normally thins local connections to make room for long-range integration happens differently. Fewer local connections are removed. The dense local wiring that was established in infancy is retained rather than cleared. The adult autistic brain is not a brain that failed to develop local connections and ended up with too many. It is a brain that developed them in the usual way and then pruned fewer of them away. Philip Shaw at the National Institutes of Health led a long-running series of brain imaging studies through the 2000s, tracking cortical development in children over years. His work showed that in ADHD, the trajectory of cortical development follows the same basic shape as typical development, but the timing is different. Regions of the cortex responsible for attention control and planning reach their peak thickness later than they do in non-ADHD brains. The gap in some key regions runs to around 3 years. The brain is heading toward the same architectural destination. It gets there on a different schedule. The behavioral consequences of this timing difference are not subtle. The cortical regions that mature late in ADHD are the same regions responsible for holding a goal in mind while ignoring competing inputs, for stopping an impulse once it has started, and for adjusting a plan when circumstances change. When those regions are 3 years behind their typical schedule, the gap between what an 8-year-old with ADHD can self-regulate and what an 8-year-old without ADHD can do is not a character difference. It is a developmental one. The same child at 11 will be closer to where the typical 8-year-old was. At 14, closer still. The trajectory converges. The difficulty is real at the point when it matters for school, for social expectations, and for early performance assessments that follow people for years afterward. Myelination is part of this. Myelin is the insulating sheath that wraps around axons and allows electrical signals to travel along them quickly and reliably. Without it, signals propagate slowly and lose strength over distance. The myelination of the brain's long-range connections proceeds through childhood and into early adulthood, and the genetic variants associated with neurodivergence include variants in the programs controlling this process. A brain whose long-range connections are myelinating on a slower schedule is a brain whose interregional communication is developing more gradually. The practical result is that the kind of integrated, multi-region processing that complex tasks require comes online later. Both of these patterns, the local wiring retention in autism and the delayed cortical maturation in ADHD, have the same upstream cause: genetic variation in the developmental programs that govern how the brain builds itself. The variants that shape myelination timing show up in genetic studies as shared risk factors across multiple conditions. So do the variants that regulate synaptic pruning. The brain does not build itself toward a single correct outcome and fail when it misses. It builds itself according to a program with many possible outputs, and the outputs vary. Some of those variations carry costs in environments built around a different expectation. Some carry advantages. Often the same variation carries both simultaneously, and that tension between cost and advantage is what produces the pattern the research has consistently found but rarely fully explained. In the early 2000s, a cognitive neuroscientist named Laurent Mottron was reviewing data from years of autism research and began to notice something the standard literature had largely set aside. The standard studies measured autistic performance on tasks designed to reveal impairment. They were calibrated to show what autistic people could not do compared to neurotypical controls, and they consistently found what they were designed to find. Motron started measuring something else, what autistic people could do that neurotypical people could not. His results were striking. In tasks involving visual search, finding a target embedded in a complex field, autistic participants were not just able to match non-autistic performance, they were faster and more accurate. In tasks requiring detection of a specific pitch or tone within a complex sound, autistic people showed discrimination abilities that exceeded neurotypical norms. In tasks requiring the systematic analysis of a long, structured sequence to find the anomaly, the same pattern appeared. The advantage was not minor or borderline, it was consistent and large. Motron and his colleagues published these findings and argued that the research field had been asking the wrong question. The question was not why autistic people fail at things that non-autistic people find easy. The question was why autistic people excel at things that non-autistic people find hard. The answer connects directly to the connectivity architecture described earlier in this episode. A brain with richer local processing within any single domain is precisely the brain you want for tasks where the fine-grained detail is what matters. One that processes incoming sensory data more directly, with less top-down smoothing, notices what is actually there rather than what was expected. Finding the single anomalous element in a long, repetitive sequence. Detecting the pitch deviation too small for a standard auditory system to register reliably. Spotting the visual target embedded in a field that a globally processing brain resolves into a unified background. These are not coincidental advantages. They are structural ones. They emerge from the same architecture that makes the sensory environment demanding and the masking cost high. The cost and the advantage are not separate features of the neurodivergent brain. They are one feature, seen from two angles, in two different kinds of environment. Motron himself went further. He had employed autistic research assistants in his lab and observed something beyond the cognitive testing data. His autistic colleagues were finding patterns in his research data that he was not finding, noticing structural features in his visual stimuli that he had not designed into them, identifying anomalies in data sets that had been reviewed and missed by non-autistic members of the same team. He published a commentary in Nature in 2011, arguing that autistic people should be actively sought out for research roles. Fields where pattern detection and systematic analytical depth were valuable were fields where the neurodivergent brain had a genuine structural edge. The neurodivergent brain was not a brain to be accommodated despite its differences. In certain environments, it was the right brain for the job. The same argument extends beyond autism. The ADHD brain, whose attention system locks onto novel and intrinsically interesting material with unusual intensity, produces a pattern called hyperfocus that is in some respects the mirror image of the attention difficulties the same brain shows in low-interest contexts. When the content engages the system, the filtering suppresses competing distractions with a grip that neurotypical attention cannot easily match. Sustained, unbroken depth of focus on something genuinely engaging is something the ADHD brain can achieve precisely because the dopamine system that regulates attention responds more strongly to interest-driven signals than to external demands. In fields where deep immersion in a problem and resistance to distraction from competing tasks are valuable, this is not a workaround, it is a structural feature. In mathematics and music, in any field where a piece of evidence can be held in sustained attention for the hours required to follow it properly, the configuration matters. The question is not whether the brain can perform these tasks. The question is what kind of environment that brain finds itself in. One that channels the focus or one that continually interrupts it. The talent asymmetry is real, but it is not a fixed property of the brain in isolation. It is a property of the brain in a particular environment. The same architecture that produces a pattern detection advantage in a quiet context with predictable sensory input produces the sensory overwhelm described earlier when placed in a standard open-plan workspace. The advantage and the difficulty are not in different parts of the brain. They are the same brain, in different environments. This is where the neurodiversity framework stops being philosophical and starts being neurological. The same developmental program that produces one set of costs in one environment produces genuine advantages in another. The brain did not make a mistake. It made a brain. When you started listening to this, you probably already had a quiet sense of whether it was about you or someone you know. Either you recognized yourself in the description, or you recognize someone in your life whose intensity of focus never quite made sense until now, whose social exhaustion after ordinary days you could not find an explanation for. The brain this episode has been about is not a rare variant. Roughly 1 in 5 people have some version of it. It has been present in every human population that has been studied across every period of history for which any record exists. It has not been removed by selection because it was never simply a cost. It came with a local processing depth that makes a person see what others look past. It came with the sensitivity calibration that picks up the signal a standard system would smooth away. It came with the pattern recognition that finds the anomaly nobody else noticed was there. The neurodivergent brain processes the world at a different frequency. The sensory environment arrives at a higher amplitude. The social world requires more explicit construction and more metabolic overhead to navigate. The environments built for the majority setting impose a sustained cost that the majority does not feel because it was built around them, but the frequency is not wrong. A frequency is not wrong, it is a setting. What decades of neuroscience have established through brain imaging and genetics and developmental biology and cognitive testing is that neurodivergence is a configuration with coherent internal logic. It is not a collection of deficits with compensating strengths. It is one architecture producing one consistent set of trade-offs in a range of environments that were built for a different architecture. The architecture did not make an error. It followed a developmental program with a different set of defaults and arrived at a brain unusually suited to some things and unusually challenged by others. Every brain is unusually suited to some things. The lines just fall in different places. The brain that finds certain environments overwhelming is the same brain that finds certain problems irresistible. The brain that loses itself in a pattern for 8 hours is the same brain that finds the social world demanding. The brain that cannot always find the expected word at the expected moment is the same brain that finds the unexpected connection no one else was looking for. This is not compensation. Compensation implies that one thing is being traded against another, that the advantage exists to make up for a deficit. That is not what the neuroscience describes. The pattern detection depth and the sensory sensitivity and the social processing difference and the masking requirement are not 4 features of 4 separate systems. They are all consequences of the same underlying architecture, a brain that weighted local processing more heavily, that kept more of its early connections, that runs predictions with a heavier reliance on incoming data and a lighter reliance on prior expectation. Every feature has the same origin. The cost and the advantage are not in different parts of the brain. They are the same configuration, read in different environments. The standard account of neurodivergence treats it as a disorder with some compensating traits. The research account describes something different. It is a configuration with coherent internal logic present in a fifth of the human population that has persisted across every human society for which any evidence exists. Not because the costs are small, they are not small, but because the costs are not the whole account. These are not separate facts about the same person. They are one fact about one brain. If you would like to see this channel continue, subscribing is the quiet and entirely appreciated way to do that.